Good morning to all. In the last class, we were discussing about what is sterilization, what are the different methods for sterilization, and we had discussed about some of the definitions like sterilization, disinfection, sanitization, what is an antiseptic, germicide, and microbiostatics preservative. After that, we discussed about what are the methods and just like a type of classification we had discussed. In that, we had seen some physical methods were there and chemical methods. In physical methods, we just briefly about what is this dry heat sterilization. In that, incineration, direct flame, red heat, hot air oven, etc. In moist heat sterilization, pasteurization, tendalization, and autoclave. In that third category was radiation and cold sterilization, use of UV rays and gamma rays, beta, X rays, ionizing reactions were there, radiations were there. And in the last category was filtration or mechanical methods. In that again there were four different methods like asbestos filters, synthread glass filter, filter candles like ceramic or Burke filled filter, membrane filter, it may be like millipore or ultra filters. In chemical methods, we had seen two different types, like one is gaseous sterilization by using this formaldehyde or ethylene oxide, or either the second one is just by directly using the chemical agents. They also may be called as by using cresol or phenol. These are one of type of disinfectants also. So this is a basic idea, a skeleton for the preparation and classification of sterilization. Furthermore, in the physical methods, when we start talking about in detail, these are the some of the methods or the processes which are going to involved by the use of physical means. They may be involving any sort of utilization of the heat in the presence or in the absence of moisture, or the application of, it may be any sort of radiations, or mechanically you are going to use any sort of filters and all. Thus, in that physical methods, the first one is dry heat sterilization. Here what happens, whatever the heat is there, in general we consider it as one of the most reliable and easy fast method for sterilization. So to kill any sort of bacteria, the killing effect of dry heat is just due to the protein denaturation. Okay. Secondly, oxidative damage and the toxic effect of the elevated levels of electrolytes. This is going to help you to improvise the process of sterilization. There are some factors which are going to influence the process of sterilization, like what are the factors which are going to influence the sterilize by heat, or nature of heat, what type of heat you are going to select, and what type of microorganisms are present in that for which you are carrying the process of sterilization. Next thing is at what temperature and the duration of the time. These are most important and most influencing factors, like nature of heat. Second thing is what type of microorganisms are present, at which temperature you are going to set up, and lastly what duration of time you are going to set, and what type of characteristics of the microorganisms they have. Okay. So whatever the time which is required for the process of sterilization is inversely proportional to the temperature of whatever exposure time is there. This can also be expressed as thermal death time, which is, it is a very less amount of time, or the minimum time which is required to kill a suspension of microorganisms at particular prescribed temperature and specific conditions. Okay, this sterilization time is directly related to the what number of microorganisms presence is there in that particular suspension, and presence and nature of spores, what category of spores are there. Okay. And furthermore, the strain and also the characteristics of the microorganisms. These are the three things on which the process of sterilization is dependent. Okay. Further, these microorganisms are more resistant to dry heat as compared to moist heat. For this reason, this process do requires higher temperature and also the period what you keep incubation period, like the temperature and longer exposure time. Whenever you are going to keep it for sterilization, at what time you, for example, in the moist heat sterilization, you do observe that at 121 to 120 degrees centigrade for 15 minutes. For why? Because when the temperature is high, obviously the duration time will be lower accordingly, depending upon the requirement, what type of, as I told you, the factors which are going to influence what type of microorganisms you are going to have, at what temperature and time. These are the most important things. So you consider in dry heat sterilization like sunlight and drying. These are some few procedures in the category of dry heat sterilization only, where you can have this sunlight and drying. Sunlight possesses UV rays, which along with the heat rays are main reason for appreciable germicidal activity. If they are passing the sunlight also, it is going to use some sort of germicidal activity. So these rays cannot penetrate through glass. Okay, this is a natural method for sterilization of water in tanks, it may be in rivers or lakes. Okay, so as you all know that drying in air has deleterious effect on many bacteria, but spores are unaffected by drying. Hence, it is very unreliable method. Just by drying or sunlight is not feasible method for the process of sterilization. Second, in this category is red heat, where it is used to sterilize these metallic objects just by holding them on a flame till they are getting red. For this reason, we do treat it as red heat. For example, from if you are withdrawing any sample from the test tube and transferring to other test tube with the help of a loop, you withdraw the sample. So before withdrawing the sample, in streak plate method, if you all remember streak plate method, I told you, take some small quantity of sample and just streak on the petri plate which is filled with the media. There also we do need this particular loop. Before withdrawing the sample, there what you are going to do, just red heat it, and after that you are going to streak. Okay. Example like inoculating wires, needles, any sort of forceps also. Okay. Just in this third category is just flaming, where the article is passed over a flame without allowing it to become red hot. Red hot. Example, I'll tell you a good thing. When you withdraw the sample with the help of this loop, so before withdrawing the sample, what you do, whatever the cotton plug is there, whatever plug is there kept closed, the moment you are going to take it out with the help of these two fingers, in the always do follow some procedures, like when you're holding your loop with the three fingers, these two fingers, with the help of the small finger and a ring finger, you are supposed to remove the cotton plug of that test tube and just pass it through the flame. Only flame, hey there, it is already there, just pass it through the flame. You should not keep it for red heat. It means that when you are the moment you are going to open, just passing to the flame, it means that no other any sort of contamination should go there. Once you withdraw the sample, again while closing, before closing this cotton plug, again you are supposed to pass it through the flame, then only close. It means that some sort of heat has passed, so entry of the microorganisms will not be there, hence no contamination will be there. Like the mouth of the culture tubes, glass slides, scalpels, needles, any sort of cover slips, etc. These items may be sometimes immersed in spirit in a tray, and the spirit is then burnt off. It is also a type of sterilization. However, it does not produce sufficient heat and destroys only most important is vegetative microorganisms. Okay, so this is about uh, the process of sterilization and flaming. Fourth one is insulation. Sure, it is one of the excellent method for rapidly destroying the materials. Example, pathological materials are there, bedding are there, animal carcasses and soiled dressings, etc. Here what happens, like the polystyrene type of materials emit clouds of dense black smoke. So for this reason, this should be autoclaved in proper appropriate containers. Okay, so these are the initial type of sterilization process. Furthermore, we will be studying about, as you all know, that in this again fifth procedure comes like hot air oven. Okay, here what happens, it's the most widely used method for process of sterilization, which is by dry heat method. Okay, whatever the modern hot air ovens are there, these are going to contain some double walled chamber of aluminum or stainless steel, which is separated from the outer case with the help of a thick layer, okay, of insulation, which is made up of fiberglass. Okay, insulation is also filled in the hollow changed door, which carries a asbestos jacket, which will be going to help you for the tight seal. Okay, so this heating is affected by with the help of electrical supply heating elements and thermostatus. Automatically this temperature control will be there in that already fitted. So the material to be arranged in a manner, so which is going to help you, like there should not be any ज़्यादा भीड़ भाड़ नहीं रहनी चाहिए, free circulation of the objects what you put, the load what you put, it should be not too heavy or too much low. Okay, if it is heavy, the process of hot installation will not be that proper, so it should be seen that it should not be overloaded. Glassware should be perfectly dry, wrapped in a craft paper before you are going to keep it in the oven. So it's also very much necessary that the oven must be allowed to cool slowly for about 2 hours before the door is open. Once you are going to get open immediately, whatever the glassware you are going to keep in that, they are going to get exposed at a temperature of 160, 170, 180 degree also. So immediately after the process of sterilization, if you just directly open the door, when you are getting that much exposed to the higher temperature, like above 150 and all, if you are immediately opening the door and taking out, so immediate exposure of lower temperature may affect some cracks may be there, since the glassware may get cracked by sudden cooling process also. So for this reason, the substances that are not heat liable and have the capacity to tolerate the temperature up to 250 may be sterilized in this hot air oven. Normally what happens, the spores as well as the vegetative cells, vegetative forms of the microorganisms are killed in 2 hours if you are going to keep it at a temperature of 160. So whatever the relation between the temperature and whatever the time which you are going to keep in the process of sterilization, in particularly when you see about hot air oven, is like if you are going to keep it for 170 degrees centigrade, 60 minutes is sufficient. If you are keeping it for 160, the same time will be more, 120 minutes. Third is you are going to maintain it at 150 temperature, the same way 150 minutes is required. Lastly, if you are going to maintain at 140 degrees centigrades, it will be like 180. However, the temperature you are going to take it to lower side, same way time will go increasing it. So this is used to particularly used for the process of sterilization of these glasswares, forceps, scalpels, scissors, spatula. Some pharmaceutical substances such as glycerin fixed oil, liquid paraffin, propylene, glycol, sulfonamides, and distinct powders such as kaolin, talc, zinc oxide, and starch. Okay, it is also not suitable for any type of surgical dressings, rubbers, because obviously higher temperature exposed you will not sustain these rubbers, whatever the surgical dressings are there. Sometimes volatile oil may be there which will not sustain this heat, or heat liable substance may be there. So hot air is a bad conductor of heat, and its penetration power is low as compared to the moist heat sterilization. Hot air ovens are commonly available in pharmaceutical laboratories for drying process and also for sterilization. This is about hot air oven. So for this, I'll close the class. Okay. So accordingly, today we had seen about what are the different types of selection process were there. How these are the physical methods. Okay, so already we have discussed about moist sterilization in not completely, but in moist sterilization we had discussed about three things: temperature above 100, at 100, and below 100. In the next class again we'll be seeing what is the moist sterilization process. Okay, thank you.